With the tissue generator, ASAP possesses the capability to characterize any type of tissue based on user-defined parameters. These tissue models could be useful to model optical systems for extracting optical properties from tissue samples in laboratory settings. In this model, we have a 0.2 millimeter slab of tissue, which is illuminated with a 630 nanometer light source. We can examine the scatter as it passes through this particular slab of tissue, as well as the absorption within the tissue using a voxel system in all 3 dimensions. And we can examine a detector which collects the light that has passed through the tissue and it has been attenuated. So let's take a look at how we could create such an optical system. Imagine we have an INR script, an ASAP script that contains a detector, a Gaussian source, and some plot, trace, and analysis information. What we're really missing here is a tissue model. In this case, a 0.2 millimeter slab of human liver tissue. Using our tissue generator, we can quickly and easily create such a tissue. What the tissue model builder requires from us is the absorption coefficient, scatter coefficient, anisotropic scatter coefficient, and thickness for each of the layers. Now here we're going to use centimeters as our unit of length for our geometry as well as for our optical coefficients. Use 630 nanometer wavelength light. Use a surface area of 10 square centimeters, a single layer. We'll use Fresnel effects, and we'll give the name test liver to our file so that we can easily find it later. And here we need to input the optical properties of our tissue, as well as layer thickness. So we need a 0.2 millimeter thickness for our sample. And we need an anisotropic scatter coefficient, absorption coefficient, and scatter coefficient for human liver. Now, these optical properties can be found in published literature. For instance, for 630 nanometer light, we can find human liver optical properties in a review of the optical properties of biological tissues, which is published online in PDF format and provides a wealth of information about different types of tissues for different animals. We can see that we have an absorption coefficient, a scatter coefficient, and a G value for human liver at this particular wavelength. And those correspond very well to our anisotropic scatter coefficient, absorption coefficient, and scatter coefficient in the slab tissue model builder. So let's input those parameters. Got 0.95 for our G value, or anisotropic scatter coefficient, 3.2 for our absorption coefficient, and 414 for our scatter coefficient in inverse centimeters. We are not provided with an index of refraction for the human liver in this particular case, so we'll assume a value of 1.45. At this point, the tissue generator has created a file that includes not only the optical properties, but also the geometry for our slab tissue. What we can do is go to our working directory, find our file, testliver.inr, and extract the information from the file by copying and pasting into the optical system that we've already created. And now we have a complete optical system, including not only our source and detector, but also the slab tissue geometry and optical properties. Let's run this file and examine the output onto a detector plane as we saw previously. We can see scatter occurring within the tissue, which is causing spreading of our source on the detector. And that's obvious from the fact that we began with a 0.25 millimeter radius source, and we have spreading as far out as around 0.5 millimeters. Returning to our 3D viewer mode, we can see we start out with a much smaller size detector, and as the light passes through the liver tissue, it spreads out to a much larger size. So to conclude, using the tissue generator, we can create slab tissue models of one or several layers that will allow us to characterize optical systems in a very realistic and helpful way in order to better understand the way that our systems work.